oxidative phosphorylation, we have one electron transport chain, okay, which is present in, so the components of the electron transport chains are present in the inner mitochondrial membrane, okay. So we have complex 1, complex 2, complex 3 and complex 4, okay. So in these complexes, these NADH and FADH releases their electron. So NADH gets oxidized and releases the electron pair into complex 1, and FADH uh, FADH2 releases the electron pair to complex 2, okay. So during the NADH electron release, the protons are pumped outside the 4 protons are pumped out inside the inner uh, intermembrane space of this mitochondria, intermembrane space, okay. And these electrons are being passed to the coenzyme ubiquinone, we say, or we can also say this ubiquinone, coenzyme Q, or you can say ubiquinone. So these electrons are passed to ubiquinone, and this ubiquinone pass the electron to the complex 3 and cytochrome C, and from the cytochrome C it goes to complex 4, where we have oxygen as electron, final electron acceptor. We have the oxygen as a final electron acceptor here. Here it gets converted into one water molecule. So the final acceptor is oxygen here, oxygen atom, and these 4 complexes having the reduction potential or the affinity to accept the electron in increasing order. So that is why the flow is maintained in one direction only, okay. And complex 2 does not pump any proton across the mitochondrial membrane, but all these complexes produce the proton, okay. And this is the in how the proton flow and the electron flow takes place, and this is the entire electron transport chain, okay. So complex 1, which is composed of iron sulfur clusters, complex 1 composed of iron sulfur clusters and FMN. Complex 2 is composed of FAD and iron sulfur clusters, and complex 3 and 4 both contains heme as a prosthetic group. Complex 3 also has iron sulfur cluster, and complex 4 has copper ions and heme, okay. So these are the prosthetic groups for these 4 complexes, okay. So the next question, if we'll see, so it is written that the prosthetic groups are cofactor involved in both 1 electron and 2 electron transfer in the mitochondrial electron transport chain are, okay. So 1 electron and 2 electron transfer here only. So as I told that NADH passes the electron to complex 1, FADH2 passes the electron to complex 2, and both will be accepted by the coenzyme Q, okay. So coenzyme Q, or also it is referred as ubiquinone, So ubiquinone, this is the symbol. Okay, so ubiquinone can take one electron at a time and gets converted into UB semiquinone. Okay, UB semiquinone, and it can take one more electron. It can take one more electron and like this, and at this time it gets the hydrogen atom and can produce this reduced, fully reduced ubiquinone. Okay, so this is what is being asked in this question: one electron and two electrons. So it can accept one electron as well as it can also accept two electrons and get fully reduced. So it is semi ubiquinone, which is partially reduced, and this is fully reduced. So this is semi ubiquinone you get after accepting only one electron, and completely reduced ubiquinone you get after accepting two electrons. So in the answer we'll have coenzyme Q, and similarly FMN also get one by one. It can accept only one electron, right? And it can accept two electrons also and convert, and these two structures it can produce or formulas. So that is the meaning of the question: which of the prosthetic groups involved in both one electron and two electron transfer in the mitochondrial electron chain transfer, while heme group can only transfer one electron, but coenzyme Q and FMN can transfer one electron as well as two electron across the mitochondrial electron transfer chain. And these uh, these two are not the correct option. Heme is the one electron transfer only, but coenzyme Q and FMN are the one and two electron transfer involved in one as well as two electron transfer in the mitochondrial electron transport chain. So that is why D is the correct answer. Okay. This is my NPTEL reference where you can read in detail about glycolysis. Thank you.